Yes, dear students of third year B Pharmacy, in the subject Medicinal Chemistry 3, the point of today's discussion is prodrugs, the basic concept behind prodrug and the applications of the prodrug design. So in this lecture, we are going to discuss about what is actually the meaning of the word prodrug, what is the basic concept and phenomenon behind the design of the prodrug, what are the applications, why we go for the designing of the prodrug, and how these prodrug concept is came in picture and how we can design the prodrugs. So this is actually included in the chapter number 2, and this is the first lecture from the prodrug design concept. Now, what we actually learn in this topic, we are going to discuss about the introductory part behind the concept of prodrug, what is actually the prodrug and prodrug design, what are the objectives behind the prodrug design, the basic concept, classification, how these prodrug concept is classified, whether it is carrier linked, whether it is drug design, prodrug of the functional groups. So how come we are going to prepare or design a prodrug depending upon the functional groups, for example ester and so on. Why we go for the designing of the prodrug, the applications of the prodrug, some of the pharmaceutical examples we are going to discuss in this particular chapter related to the prodrug, and what are the recent searches and recent work is carried out with respect to the prodrug, conclusion and some of the references. Now, introduction of the concept of prodrug. Before we actually discuss this slide, I need to clarify some of the history points related to the prodrug. Basically, almost all the drug possesses undesirable physico-chemical and biological properties, because we all know drug is going to have its own some physico-chemical and biological property. Drug candidates are often discontinued due to some of the issues of poor pharmacokinetic properties or high toxicities. So some of the drug molecules are going to have poor pharmacokinetic properties, for example solubility, dissolution and so on, and some drugs have the presence of some functional groups which will offer high toxicities in our systemic circulation. Their therapeutic efficacy can be improved by eliminating those undesirable properties while retaining in our body. So some drug molecules are having poor pharmacokinetic properties, and some drug molecules, although they are good good pharmaceutical agent, but they have toxic related effects in our body. This can be achieved through biological, physical and chemical means. So the biological approach of altering the route of administrations. If some drug molecules are going to give the poor pharmacokinetic properties, so we can go for the another route of administration in order to alter the biological properties. When we are going to alter the physical approach, we are modifying the design of the dosage form. If we are going to change the physical properties, so we will go for the modification of the design of the dosage form, and the best approach in enhancing the drug selectivity while minimizing the toxicity is the chemical approach. So we can alter the drug through either biological approach, physical approach or by the chemical approach. If we are altering the biological approach, we are going to change the route of administration. If we are going to alter the physical approach, we are going to change the dosage form. And if we are going to alter the chemical approach, then we will go for the preparation of the prodrug. Now, what is basically a prodrug? What is the definition of the prodrug? The prodrug is a drug product that is inert in the expected pharmacological activities. Now basically the prodrug is inert in nature and going to have in the expected pharmacological activities and must be transformed into a pharmaceutically or pharmacologically active agent by metabolic or physicochemical transformation. Now just now we have discussed that drug candidate may go for the biological change, may go for the physical change and may go for the chemical change. So it is transformed into the pharmacologically active agent either by metabolic or physicochemical transformations. So prodrug can be found in nature such as several phytochemicals and botanical constituents and endogenous compounds. So drugs may present in the phytochemicals, biological constituents, or some drugs are going to have the endogenous substances or the compounds. They can be created by synthetic or semi-synthetic methods. Now these drug molecules can be created by either synthetic or semi-synthetic methods, either deliberately or by accidentally through the drug creation. So this is a basic concept behind prodrug approach. We are altering the pharmacological active agent by either metabolic or physicochemical transformation. And in view of the same, in 1958, Albert introduced the prodrug concept for the first time. So the first time who has prepared the prodrug concept or who has introduced the prodrug concept? Albert in 1958 introduced this concept first time, that is selective toxicity. So this term include any inert compound that undergoes in vivo biotransformation. Biotransformation is nothing but the metabolical changes. Now in the example of such compound is in front of us, that is methenamine. Now this methenamine was discovered in 1899 by Skring as inactive prodrug which delivers the antibacterial formaldehyde. This methenamine, if it is coming in contact with the acidic pH, it will change its structure and the product obtained is a formaldehyde plus ammonia. Now this formaldehyde is going to have the antibacterial properties which is useful in the treatment of urinary tract infections when transported to the urinary bladder. If this particular compound, if it is transported to urinary bladder, it is going to have the effect against urinary tract infections. It becomes acidified to provide a medium in which formaldehyde is generated. So methenamide, which is inactive as such, and if it is coming in contact with acidic pH, the product obtained is formaldehyde and having an antibacterial actions. Now what is actually a prodrug and how we are going to go for the design of the prodrug? Now prodrug basically is a pharmacological substance which is administered in an inactive form. Now this particular drug is administered in our body in the inactive form, but once it is administered, the prodrug is metabolized in vivo into an active drug within the body through some of the metabolic processes. Now these metabolic processes involves hydrolysis of an ester form of the drug and so on. So such a type of simple procedure or simple chemical reactions gives us the active form of the drug. Sometimes drugs are designed to make use of metabolic processes in order to generate their active forms. What happened? Sometimes the drugs are purposefully designed to make the use of metabolic processes in order to generate their active form. This is done in order to improve some selected properties of the molecule, such as if we are going to alter the water solubility or ability to cross the biological membrane temporarily. So in order to cross the biological membrane, we are going to go for the design of the prodrug. Basically, the drug is administered in the inactive before its administration, but through some of the metabolic biotransformation processes in the liver, drug become active after the metabolism. Now, what are the basic objectives that we will go for the design of the prodrug? There are main objectives of the prodrug are to bring the active drug to their respective active site. If the drug is in its active form unable to reach the active site or target location, so through prodrug approach we will reach, or we can bring the active drug to their respective active site, as well to provide the desired pharmacological effect while minimizing adverse metabolic and toxicological events. So it is going to provide the desired pharmacological effects while minimizing the adverse metabolic and toxicological events. To improve the clinical and therapeutic effectiveness, it is going to be improve the clinical and therapeutic effectiveness of those drugs which suffer from some undesirable properties that otherwise hinder their clinical usefulness. So this prodrug approach also improves the clinical and therapeutic effectiveness. It is also used to avoid the practice of clinically administering two drugs in order to enhance the pharmacological activity or prevent the clinical side effects. That means we can give two drugs through co-administering two drugs in order to enhance the pharmacological activity and prevents the clinical side effects. So these are basic objectives. Some are pharmaceutical objectives, some are going to alter the pharmacokinetic objectives, and some are going to alter the pharmacodynamic objectives. So overall discussion of this is, some will alter the pharmaceutical, some will alter the pharmacokinetics, and some are going to alter the pharmacodynamic objectives of the prodrug. Now, what is the basic concept behind the designing of the prodrug? Now, the drug is a moiety, is the prodrug which is typically pharmacologically inactive as such. Now, this inactive drug through prodrug approach we are converting into the active, but limitations of the parent drug that prevent the optimal biopharmaceutical or pharmacokinetic performance. Now what happened? The main objective behind the prodrug design is, parent drug prevents the optimal biopharmaceutical or pharmacokinetic performance of the drug molecule as such. So through prodrug approach, what happen if this, now for example this drug unable to cross this barrier to reach the target location. So the drug unable to reach the target location to show their pharmacological response. So what we did, or what changes we are going to make? Drug along with the moiety, that is converting the drug molecule into the prodrug, can change some pharmaceutical, some pharmacokinetic properties and some physical properties in such a way that this drug plus moiety concept can easily cross the barrier to reach the target location. Now, what is basically this pro-moiety? What this pro-moiety is doing? Pro-moiety is nothing but a functional group which is used to modify the structure of pharmacologically active agent to improve. What this prodrug is doing? It is improving the physicochemical properties, biopharmaceutical properties, or sometime going to increase the pharmacokinetic properties. So this drug along with the pro-moiety complex can easily cross the barrier through changing physicochemical, biopharmaceutical or pharmacokinetic properties. We are going to get enzymatic and chemical transformations. After that, the drug reaches the target location. This pro-moiety is again recirculated for the entry of another drug molecule. Now, classification of the prodrug. There are varieties of ways to classify prodrug, which includes carrier-linked prodrug, then bioprecursors and photoactivated prodrugs. So there are basically three types. First is carrier-linked prodrug. Now, what is the meaning of the carrier-linked prodrug? This is the first class of classification of the prodrug. This carrier-linked prodrug is nothing but simple prodrug that contain an active drug which is linked with the carrier group, and that carrier group, after when the drug reaches the target location, it is removed enzymatically. So that means nothing but the drug product, the prodrug which containing an active drug which is linked with the carrier group. Now this carrier group simply carries the drug molecule from the biological or carrier membrane. Now the carrier group must be that carrier group which is going to be provide the or altering the for physicochemical properties. It must be non-toxic, biologically inactive when detached from the drug molecule. When the carrier group is detached from the drug molecule, it must be non-toxic in nature and it must be biologically inactive in nature after detaching the drug molecule to the target location. So how come we are going to represent the diagrammatically? That is active drug plus the inner carrier molecule. They are going to form a complex. That is chemical prodrug formulation, because we are changing the physicochemical properties. So what happened? Chemical prodrug formulation reaches the target location and it will be cleaved in vivo into the circulation. And they are going to cleave the covalent bond which is formed between the drug molecule and the carrier molecule. Now, this carrier-linked prodrug concept or the class is further divided into 4 classes. Now, this carrier-linked prodrugs are further divided into 4 classes. First is double prodrug or cascade latentiated prodrug. Now, in this, that is double prodrug concept, where prodrug is further derived in a fashion such a way that only enzymatic convergence to the prodrug is possible before the latter can be cleaved to release the active drug. What is the double prodrug concept? In which the prodrug is further divided in a fashion such that only enzymatic conversion to prodrug is possible before the latter can cleave to release the active drug. So in the first step, only enzymatic cleavage is happening and later on active drug is released to the target location. Next is macromolecular prodrug. The meaning of the word macromolecular prodrug is where macromolecules like polysaccharides, dextrans, cyclodextrins, proteins, peptides and polymers are used as a carrier. In this case, some macromolecules having a big structures, big sugar molecules like dextrans, cyclodextrins, peptide, proteins and polymers are used as a carrier molecule. Now next is site-specific prodrug, where the carrier acts as a transporter of the active drug to a specific targeted site. In this case, the carrier is act as a transporter of the active drug to the specific target location. This is site-specific prodrug. And finally, and the last class is mutual prodrug, where the carrier used to another biologically active drug instead of some inert molecule. Mutual prodrug is nothing but the carrier is going to have a pharmacological actions, and this carrier molecule, which is biologically active drug instead of some inert molecule. So these are the basically very important 4 classes of the classification of carrier linked prodrug, and the classes are double prodrug concept, then macromolecular prodrug, site specific prodrug, and mutual prodrug. Now the second class of carrier linked prodrug is bioprecursors. Now bioprecursor is a compound which is metabolized by molecular modification into a new compound that may itself be active or further metabolized to an active metabolite. The compound which is metabolized by through molecular modifications. After molecular modifications, we are going to get a new compound, and that may itself be active or further metabolized to an active metabolite. So what is actually a bioprecursors? The compound that is metabolized by molecular modifications, that means our phase 1, phase 2 reactions, into a new compound, and this new compound may itself be active or it may be further again metabolized to get an active metabolite. This prodrug does not contain carriers but ready upon metabolism to induce the necessary functionally active species. So when the prodrug is going to give the active drug, the drug does not contain any carrier group but ready up for on metabolism to induce the necessarily functionally active species. Bioprecursor prodrug rely on phase 1 and phase 2 reactions of our biotransformations. Now we all know phase 1 and phase 2 reactions. Phase 1 reactions of our metabolism are generally oxidative and reductive activation species, unlike hydrolytic activation of the carrier linked prodrug. They metabolized into a new compound that may itself be active or further metabolized to an active metabolite. For example, amine to aldehyde, aldehyde to carboxylic acid, inactive prontosyl to sulfonamide. Now we all know we can convert the amine functional group to aldehyde and aldehyde through oxidation into the carboxylic acid. So these are some of the examples of metabolism or biotransformation reactions for conversion of inactive metabolite into active. That means, for example, first of all, amine conversion to the aldehyde. Still the compound is not that much active as such. So further the aldehyde is converted into the carboxylic acid. So this type of concept is bioprecursor. Again, some of the example, that is conversion of inactive prontosyl to the sulfonamide. Now conversion of prontosyl to the sulfonamide, that is prontosyl undergo through metabolic changes, it is converted into the sulfonamide, which is basically an antibacterial agent. Now what happened to this prontosyl molecule? This prontosyl molecule in our body, it is going under for the metabolic reduction, and through metabolic reduction it is converted into the sulfonamide moiety, which is actually going to have a good antibacterial action, whereas the prontosyl molecule, as such, it is inactive in nature. So this is a prontosyl combined nucleus of azide. A type of nitrogen bond is there, and in which we are going to cleave this bond, and we are going to get the sulfonamide moiety, which is going to have the active antibacterial properties. So this is a basic concept behind the prodrug design. Now, conversion of some of the functional groups in the case of prodrug concept. In this that is prodrugs of the functional group. So through the prodrug approach, we are going to change the functional groups. Those are various types of functional groups are present in different therapeutic agent. Now we all know drug is going to have the presence of minimally it is going to have the presence of 4 to 5 functional groups. Now these functional groups react with the other functional groups of non-toxic pro-moiety to form the prodrugs. These functional groups may react with another functional groups and forming a prodrug. Now here we will discuss example of first functional group, that is ester. Now groups like carboxylic acid and hydroxyl group can easily undergo esterification reaction. Now we all know acidic functional group and alcoholic functional groups will come together and there is formation of an ester bond. So they will easily go or undergo for the esterification reaction, and bioavailability of the drug can be improved by ester formation. If the carboxylic acid and alcohol functional group go for the ester formation, they will increase the bioavailability of the drug. And for example, the enzyme esterase, which is present widely in vivo, can easily break up the linkage, that is ester linkage, at the target organ, so that the targeted delivery is achieved through formation of the ester bond between the functional groups like carboxylic acid and hydroxyl group. And the example of such drug is thioester of erythromycin. Another example is palmitate ester of clindamycin. And this ester forms of the drugs, whether it is of erythromycin, that is thioester of erythromycin, or palmitate ester of clindamycin, will easily go for the enzymatic cleavage reaction at the target location through the enzyme, that is esterase. In the presence of water molecule, we will get the active form of the drug through the enzymatic cleavage reaction. So this is a first example of the first functional group ester. Through the breakage of this ester bond by the active in vivo enzyme, that is esterase, we all get the active form of the drug. And the example of such type of drug is enalapril. Enalapril is also an ester form of the drug, and after metabolism we will get the enalapril. Next example of the functional group is amide, in which the utility of the N-acyloxy alkoxy carbonyl derivative is limited due to the resistance to undergo enzymatic cleavage in vivo. So that is N-acyloxy carbonyl functional group is going to have the limitation due to the resistance to undergo enzymatic cleavage in vivo. This functional group is unable to cleave easily. However, certain activated amides are chemically liable, and also certain amides formed with the amino acid may undergo enzymatic cleavage. For example, alpha glutamyl derivatives of dopamine, that is L-dopa, and N-glycyl derivatives. Now we all know dopamine is the neurotransmitter, dopaminergic neurotransmitter in our body, having the presence of amide functional group, which is metabolically converted, that is L-dopa and N-glycyl derivatives. Now next example is prodrugs of amides, imides and ureides. The N-hydroxyl derivative of amides or imide type compounds are more water soluble than parent molecule. amide and imide derivatives of the drug molecules are going to have the these compounds are more water soluble than the parent compounds. Now, by replacing the proton bind to the nitrogen atom, if we are going to replace the proton groups to the nitrogen atom by hydroxyl methyl group, substitution of the replacing proton, that is hydrogen, through nitrogen atom by hydroxyl methyl group, either intra or intermolecular hydrogen bonds in such molecules may be increased, resulting in the decreasing in the melting point and increases water solubility. So change in the functional groups by replacing proton through by nitrogen atom, the water solubility is increased. That means this is the mechanism for the decomposition of N-hydroxyl methyl derivatives. Now the next approach is of preparation of through functional group changes prodrug is ring formation derivatives. Thiamine quaternary ammonium compounds, like now some drug molecules have the presence of nitrogen atom and they are going to form the quaternary nitrogen compounds like hadantoin, barbituric acid, can undergo ring opening and show in vivo pharmacological properties, that is ring formation derivatives. Then next is glycoamide esters. These are bioavailable products of carboxylic acid, that is ester of benzoic acid. Next is carbamide. They exhibit restricted distribution in the body. Carbamide do not have any specific enzyme for hydrolysis. However, enzymes such as esterase can hydrolyze the carbamide derivatives, for example, into carboxymethylphenyl ester of amphetamine. Now this drug amphetamine is basically a carboxymethylphenyl ester of amphetamine, that is carbamide. Other approaches used in the formation of prodrug are phosphamides, glycosides, ethers and ketal molecules. Now, why prodrug? Why will go for the design of the prodrug? Why is the because prodrug is going to improve the membrane permeability, improves absorption and distribution, improve solubility, alter metabolism, alter toxicity and alter eliminations. Now, some of the applications of the prodrug concept and prodrug design is going to have the pharmaceutical as well as pharmacokinetic application. The pharmaceutical applications are improving the taste, improvement in the odor, reduction of irritation, reduction of the pain and injections, enhancement of the drug solubility and dissolution, enhancement of the chemical solubility stability of the drug. Then next, pharmacokinetic applications are enhancement of probability, prevention of presynaptic metabolism in the first pass effect, prolongation of duration of the actions, reduction of the toxicity, and the drug is given at the target location, that is a specific site drug delivery. Now, for example, ester through enzymatic cleavage esterase, we are going to get the active compound, that is active form of the drug. Enalaprillase, monoethyl ester of enalaprillate, was first discovered as an inhibitor of angiotensin converting enzyme and used to treat the hypertension. Now this enalapril is basically used to treat the hypertension. Due to its high polarity, it was not orally bioavailable and thus needed to be administered by the injection, that is enalapril. Now, conclusion behind the design of the prodrug is why we go for the designing of the prodrug. What is the concluding remark of the prodrug design is prodrugs are inactive compounds which are converted to the active drug in the body. Prodrugs were designed to improve the pharmacokinetic and drug delivery properties. Esters are commonly used as a prodrug to make drug less pora, and the ester bonds can be easily processed. The nature of the ester can be altered to vary and rate of hydrolysis. Now these are some of the references which I have used for the preparation of this particular presentation of Prodrug basic concept and design. Thanks for listening, and the more videos of such type of lectures are there in my YouTube channel. Please do subscribe for the same. And the name of the channel is T.Y.B.Pharm Pharma Chemistry Lectures all together. Here you are going to get the lectures of all the subjects which are there, subtypes of pharmaceutical chemistry subjects. So this is a link of my YouTube channel. Thank you all. Please do subscribe, and thank you all. Thank you.